Hello and welcome to Chapter 16 on development, stem cells, and cancer. Let's get right into it. Now, if you think about multicellular organisms like us, we all start as a single cell. And through cell division and then differentiation of certain cells into tissues, we end up with this multicellular organism that has different tissues that have different functions. And we think back to gene expression. It's all orchestrated through these differences in gene expression, because ultimately the genetic information in all those cells is the same. Now, we know a lot about how development works by studying model organisms. Some examples would be fruit flies, so Drosophila. This is the example here you see on the bottom right. And then on the left, bottom left here is the example of a zebrafish. These are just two kinds of model organisms that are great for studying development. So starting with that single fertilized egg, we see cells go through a process of differentiation into tissues and then organs and organ systems and the whole organism. And how that all happens is very, very coordinated, and the cells interact with each other, sort of cross-talking to determine who's going to do what and what's going to become what tissue layer, etc. And so the steps in this process are cell division, cell differentiation, and morphogenesis. So cell differentiation, well, first, we all know about cell division. That's just the cells dividing. Cell differentiation, this is where we see cells that, say, like stem cells that don't have, that haven't differentiated, that can become different kinds of cells, basically changing or becoming specialized into certain cell types. So here in this figure, we see stem cell here in the middle. This has the potential to differentiate into all these different kinds of cells, and then those come together as tissues. So morphogenesis, that's that next step. This is the actual shape. So morphology is shape, right? So morphogenesis is the organism taking shape. So how differentiation occurs, it can happen in a few ways, and we'll look at some of the signals that determine what a cell is going to do. And we'll start with cytoplasmic determinants. And so these would be things, signaling molecules basically, that are already within a cell. And this, we think back to that original zygote. So at fertilization, that egg cell and then zygote may already have signaling molecules or even RNA that will code for proteins that determine what's going to happen after fertilization. These things are not distributed evenly throughout the cell, so they have an uneven distribution, and that creates sort of polarity in say the next cells that are divided here. So we see this in this figure. We have these cytoplasmic determinants, just colors, little triangles and circles. And so when this cell undergoes this mitotic division, you see the two daughter cells, the two-celled embryo here, having different amounts of these cytoplasmic determinants as this cell division has happened. Here, this guy on the left, or sorry, on the right, gets all the green triangles. This one on the left gets mostly the little round circle cytoplasmic determinants. And so this is going to lead now to differences in gene expression in these two different cells, again, depending on what these signaling molecules are, or if they're RNAs, these are going to express different proteins. And so that will determine what happens to these two cells, and ultimately becomes four, and so on. So those internal cytoplasmic determinants are one source of information. And another source of information is external, and this is called induction. So when cells are receiving these external signals from a neighboring cell, think back to like paracrine signaling, if you remember that from cell signaling. So para means next to. So one cell is releasing a signaling molecule, and the cell next to it is receiving that signal and being induced, right? So this gets a signal, okay, turn on this set of genes. And so this is also really important, as we see as this embryo is now at the 32 cell stage. And these cells are sending signaling molecules to these neighboring cells because these cells are going to ultimately be doing different things. These guys are releasing the signals, these guys are receiving them, and so on. So as these signals are being received, either cytoplasmically or from external cells, so through cell signaling, this is determining the cell's fate. And so sets of genes get turned on. You're going to become a liver cell, for example. And this sets the cell on that path to differentiation. So this determination, which is the genes being switched on, receiving those signals, that precedes differentiation. And so once a cell starts down that path towards differentiation, it will still continue down that path. And we know this from experiments where we say take a cell that's already started that determination process or differentiation process. So it's already been determined what it's going to be, but not differentiated. It will develop according to that original set of signals, even if we move it somewhere else. All right, so with that in mind, what might you look for? What's the very first evidence of differentiation in cells of an embryo? So very first evidence, what would you look for? All right, you had a few choices there. One was just seeing that the cell looks different. That would be sort of the later stage of differentiation. So the cell is ultimately taking the shape and structure or whatever form that it's going to take. But the very first evidence would be the reception of those signals and those genes being turned on. So that's what you'd look for, is the presence of certain messenger RNA that's going to determine what that cell will become. And so that's what we would see. We see the switching on of sets of genes. And there are a wide range of what are called master regulatory genes. So a certain signal will turn on, a gene that produces a transcription factor, let's say, that switches on a whole bunch of other genes that are all tied together that are important for the differentiation of a certain cell type. And we see that here in this example. We see our embryonic precursor cell, and there's this master regulatory gene, MyoD. When this gets turned on or transcribed, so we start to see this cell, it's in determination here. It still looks like an embryonic cell, but at this point, that gene expression's already started to happen. This MyoD protein's been produced. It's a transcription factor that's going to switch on a whole bunch of other genes that will ultimately cause this to differentiate into a muscle cell. Now, morphologically, this looks like a muscle cell now, but it was already determined back here that that's what this was going to become. And so these master regulatory genes are really important because they switch on a whole bunch of other genes and that set the cell along its course in differentiation. All right, something else that we see in development, which maybe if you're first learning about this, maybe seems counterintuitive, but that some cells are programmed to die. And we've talked about this before when we talked about the cell cycle, that if something goes wrong with the cell cycle, of those checkpoints, the cell may be basically destroyed at that point if there's an error. And that's not just a random thing where the cell explodes. It actually undergoes this process called apoptosis, which is also known as programmed cell death. And this is not just because DNA is damaged or there's a problem, but it may be actually critical for development. So what does apoptosis look like? So it's a very, very much a programmed cell death. If a cell were to die and just, say, explode, all that material from within the cell now is spreading around against the neighboring cells, and that may cause damage to those neighboring cells. Or in a grown organism, the immune system might react to that and cause some damage to those neighboring cells. So that wouldn't work very well. So what happens in apoptosis is this very systematic process where the DNA is broken up. The cell starts to sort of bud off as pieces and parts of it are still encapsulated within membrane, but are moved away from that area as the cell fragments. And then that material can be scavenged and reused, and there's been very minimal damage to the surrounding cells. So where do we see that in development? Here's a really cool example here. So here's mouse paw development and some really neat images. So here we have the interdigital tissue. That means the between the fingers, the developing digits. And so how do those clefts form, those spaces form? Here we see through a labeling experiment, cells undergoing apoptosis at those spaces. So these cells are dying away and actually creating the space between the digits. So that apoptosis isn't just a bad thing where, the cell is dead or there's a mistake been made. It can be, but it's also really critical in development in something like this. Another really important area of development is pattern formation. This idea of the body plan taking shape. And it's really important when this is happening that those developing tissues are developing in the right places and relative to one another. And so there is a right and a left and a back and a front and a head and a tail, and that all of these things are working in concert and they sort of quote unquote know where they are in this developing organism. And so these major axes are critical. The other thing that's important in pattern formation is just positional information. Not just along those axes, that's also positional, but say in a developing limb like we see here. So there are molecular cues that are occurring for say this wing development in this example, where we have this limb with the different components developing in the correct places relative to one another. Now, this kind of positional information and pattern formation has been studied at length in a model organism like Drosophila, again, many others as well, but Drosophila is sort of a famous one for studying this, especially in the early days. So we know a lot about how is it determined, say, what becomes the head versus what becomes the abdomen in this case, or tail, you can think of at the other end. First, we can look at, say, the life cycle of Drosophila here. So Drosophila, they're insects. They start as an egg, and then we have this mature unfertilized egg. And already at this point, we start to see some endedness to this cell. The egg becomes fertilized and then undergoes development. So in the unfertilized egg, there are already those cytoplasmic determinants like we talked about before, and they're unevenly distributed within the egg. And that's going to help determine what it becomes the head and what becomes the tail in or abdomen in Drosophila. And those cytoplasmic determinants are really important for influencing a variety of genes, but in particular, the homeotic genes. These are really crucial for controlling pattern formation in, say, fruit flies, or also vertebrate animals. Here we see mouse. We see this color-coded, and we can see along the length of this organism, whether it's a fruit fly or a mammal, we see different groups of these homeotic genes being switched on, and they're going to determine what happens over here at the head end, what's going to happen in the middle, what's happening down in the posterior. Again, these are those master regulatory genes like we saw in that muscle cell differentiation. Now, there's many different sets of these master genes. How do we know about this? So variety of scientists basically researching genetic mutants that don't undergo proper development. This is a way to identify genes that are associated with development for when it does work. And so this Nuslin, Volhard, and Weisshaus studying Drosophila identified embryonic lethals. And so they're looking for genetic mutations that don't allow development in the fruit flies. And through that, they identified 120 genes that are essential for normal segmentation in fruit flies. So there's, again, a lot of genes involved in this. and where those genes are turned on and what part of the organism is very crucial. And so let's come back to that very early development in the egg, those cytoplasmic determinants. One type are called maternal effect genes. So these encode cytoplasmic determinants. So those maternal effect genes create those cytoplasmic determinants. Those are going to determine what's the head and what's the tail in the body of a Drosophila organism. What's really interesting here is if you have mutations in these genes of the mother, then the phenotype is seen in the offspring, right? Because these are already there as cytoplasmic determinants in the egg of that offspring. So even if that mutation wasn't in that particular offspring, the mother has passed those cytoplasmic determinants along to the offspring. Another term you'll hear for this are egg polarity genes, basically because they determine the endedness of the eggs. But again, they are cytoplasmic determinants as a result or produced from maternal effect genes. An example of this is called bicoid. So this is one maternal effect gene, and this determines the head end of a Drosophila embryo. And we know if we knock this out, you'll have an egg that will develop that has basically two tails. It doesn't form a head end. So we know bicoid has to be there to determine where the head of that developing Drosophila goes. And we could see that here using a staining technique for the bicoid messenger RNA. Where do we see it concentrated? Here at this anterior end of the egg. And over time, you see it diffuse. So after fertilization, this bicoid messenger RNA starts to be translated, so producing the protein, and it will diffuse a little bit further back here, but still mostly concentrated toward the anterior end. And this will determine that this is going to be the head end of this organism. So bicoid is also known as a morphogen because we know that this is just a critical gene product for determining the morphology and establishing the axis, in this case, head versus tail. We can look in a little more detail here at bicoid initially. So at fertilization here, we see lots of messenger RNA really concentrated down at the anterior end. And then as those proteins are translated, they diffuse. So they do diffuse a little further back in the egg, still more highly concentrated at the end here. And this is that gradient hypothesis, the idea that it's not just an all or nothing thing, but there's a gradient. There is some of that bicoid protein a little further down, but it's mostly concentrated at the head end of what will become the head end of this organism. We also know through experimentation, if you inject bicoid at different places in a developing embryo, that will result in the formation of anterior structures. So this bicoid gene research is critical for a lot of reasons, but here are three highlights. This idea that a certain protein can be crucial for those early steps in pattern formation, head to tail, dorsal, ventral, left and right. They may also be present before even fertilization has occurred, and this highlights the role of the mother in embryonic development. So those maternal effect genes, those things that are already there as cytoplasmic determinants that are produced from the mother. And the other thing that we saw here, and that's crucial for that polarity, is that these things may be associated with a gradient, like bicoid is, it's more concentrated at one end or one part of that developing embryo, and that determines a certain component, like the head of, say, Drosophila. All right, so what if there's a mutation in the bicoid gene? What would a mutation most likely affect? Right, so this is going to have the biggest impact on what's going to become the head and what's going to become the tail, or anterior versus posterior of that developing embryo. All right, so let's move on now and talk about cloning organisms. And so these things are all connected, because when we think about cloning organisms, we have to look at the idea of stem cells. And so stem cells are these undifferentiated, undetermined cells that can become differentiated into cells. And there's a lot of interest in creating clones to generate stem cells that can be used, say, for medical treatments, for example. And so cloning, what's happening is we're taking individual, say usually a differentiated cell, and from that creating another organism. So clone individual, or clone in general, is just things that are genetically identical. When we're thinking about cloning, we're taking that cell and creating another organism from it, again, that is genetically identical. So we can take an example from the early days of cloning here with plant cloning. So this is work done by F.C. Stewart back in the 1950s, taking single differentiated cells from roots of carrots. The roots, of course, is the actual carrot there, and putting that into a cell culture medium. And so those cells would divide and ultimately form a complete single plant. So this is really interesting, because those differentiated root cells, they weren't locked into that form. So that kind of suggests in some cases that differentiation can be reversible. And so cells that can, say, start like this from a single cell, producing, say, a whole organism with all its differentiated tissues, those are called totipotent. So toti kind of means everything or all. They can form, they have the potency to form, or potential to form, any kind of cell. All right, so if entire plants can be cloned from an individual somatic cell, like that root cell, what does this demonstrate? All right, let's look at cloning animals now. This is a little bit more difficult than with plants, because those cells that are differentiated, usually they can't sort of just go back and be reset and become then any other kind of cell. So something we see with animal cloning is something called nuclear transplantation. And so in this case, we're taking a nucleus from say, an adult cell, preferably from not a fully differentiated cell. And that's we can see the difference here in this frog experiment. So here's this frog cell, the nucleus is removed, the egg cell here, the nucleus is removed, and we're going to put a nucleus in from, say, this frog embryo. So this is not a fully differentiated cell. When we do that, we get most developing into tadpoles. So this sort of early nucleus works okay for nuclear transplantation. But if we take a nucleus from a fully differentiated cell, like this intestinal cell, some development occurs, but it never makes it to the tadpole stage in this case. So it's important that you're getting a nucleus from a cell that is not fully differentiated. The further along you are in development, say like in this tadpole taking this intestinal cell, the more sort of restricted that nucleus is in terms of it being fuel for cloning. So some of you maybe heard of this. This was one of the early experiments in cloning in mammals. Here's Dolly the sheep here. That was a result of nuclear transplantation, like we just talked about, from a differentiated cell. And so this is like that frog example there. We're taking a differentiated cell, not an early developing cell, and produced this new sheep, which was called Dolly. Now, interestingly enough, this did work, but Dolly did have a premature death, arthritis, and that was kind of interesting because this makes us wonder, like, what was, say, in that nucleus of that differentiated cell that we used for the starting point of our cloning? Is there information there that kind of set the lifespan of those cells at a later point because it was already a differentiated cell. And that nucleus wasn't really reset for like, this is a zygote, this is a single cell, but it was a differentiated nucleus. Now, this kind of work has been done several times in the last 20 plus years. Here's another interesting example here. CC, for carbon copy, this cat here on the right, this is CC here on the right. But CC was different in hair color pattern here. So we see the parent cat has patches of orange fur, but CC did not have orange patches of fur. And so this kind of shows us that the stages in development don't necessarily proceed exactly the same way. And that clones aren't necessarily identical, although, at the gene level, it seems that they are. They're not necessarily identical in their behavior or appearance to their parents, relative to their parents. And if we wonder why that might be, think back to this idea of epigenetics, where we see differences in methylation patterns as a mode to regulate gene expression. The individual cell that's used for those nuclear transplantation experiments, it has acquired methylation patterns during the life of, say, that cat or donor sheep that are going to be passed along to that recipient clone. And so this is probably one of the critical hindrances or stumbling blocks in cloning. And there really needs to be a way to reverse any of those epigenetic changes to sort of reset the clock for that particular nucleus to the point where it is like a totipotent nucleus that can be transplanted into that developing embryo. And so this loops us back to the idea of stem cells. So a stem cell, as we've talked about, it's an undifferentiated, unspecialized cell. They have the ability to reproduce indefinitely. So this is really interesting from a research perspective or medical treatment perspective. And they have the ability to form a huge range of cells. So if you're starting with a stem cell, maybe you need to replace some muscle cells or whatever kind of cell that you're interested in from a, again, maybe medical treatment perspective, that they have that potential to become any one of those cells. And so here in this figure, we see stem cells dividing, making other stem cells. Here along this path, we see the cell beginning to differentiate. So determination's happening here, and that can then become all these different kinds of cells through differentiation. So generally, there are two kinds of stem cells, embryonic and adult stem cells. So early embryonic cells in animals, those are pluripotent, so not totipotent, which is basically that can become anything, but they're pluripotent, meaning they can become, there's a plurality of things that they have the potential to become. They can reproduce indefinitely, and these will be the basis of forming all these other kinds of cells that ultimately differentiate into these different cell types that we see in different tissues. Adults also have stem cells, but these are limited in what kinds of cell types that they can differentiate into, rather than, say, embryonic cells that can differentiate. In this case here, we see them being able to differentiate in all these different kinds of cell types given different signals, whereas these adult stem cells, say from bone marrow, these can produce blood cells, but they can't produce nerve cells or liver cells. So they're limited in what they can form, but still, again, still potentially very, very useful. And so there's a ton of interest in stem cells from a medical perspective, research perspective, and the potential ability to take a differentiated cell, say like this skin fibroblast cell, is there a way to reprogram it and sort of, reset that clock so that it can act like a embryonic stem cell? And then we can use it to create all kinds of cell types. And so there is a way to do this in the lab. These are called induced pluripotent stem cells. They're induced because using a viral vector, specifically a retrovirus, this is a kind of virus which will deliver its genetic information and integrate into the genome. And so we can do this very specifically, and this will reset that nucleus to sort of stem cell mode. Now, these aren't exactly the same as embryonic stem cells, but they do perform very similarly to embryonic stem cells in terms of how they function. And again, there's a lot of potential for these using these in medical treatments. And just two examples of where we could use these IPSs, induced pluripotent stem cells, would be in the case of Parkinson's disease or type 1 diabetes, where you're replacing lost cells with these programmed cells that will replace pancreatic or nerve tissue in the case of diabetes or Parkinson's, respectively. And ideally, you would take that individual patient's own cells and reprogram those and to replace these destroyed or degrading cells. And a final note on these IPSs is this idea of taking a differentiated cell and not going back to the pluripotent stage, but actually being able to take a differentiated cell and then making it into another differentiated cell, maybe cells that are not even closely related in the same lineage. This would be a huge step forward in terms of using this as a medical treatment. Now, I will say these last couple of things here really are more in the lab stage at this point in terms of their broad use. We are a ways away from that, but it is very exciting to see what the future holds with the use of these kinds of stem cells. All right, so here's a question for you. So researchers using adult stem cells, remember adult stem cells, comparing them to other adult cells from the same tissue. What statement describes a likely finding here? Which of these makes the most sense? All right, so you would expect different gene expression patterns. And another thing you might expect when you're comparing a stem cell versus an older cell, differentiated cell, would be differences in methylation patterns. So that's where the answer comes from here, is that older cell is going to have probably more methylation that is regulating that gene expression. All right, let's finish up this chapter with how gene regulation relates to cancer. And we've seen this before, this idea when we talked about the cell cycle, that cancer is a failure to regulate the cell cycle. And there are two kinds of genes associated with cancer. They're called proto-oncogenes and tumor suppressor genes. So these proto-oncogenes we can look at first. They're proto-oncogenes because they have a normal function in cells. They're often growth factor genes, or they're really important in early embryonic development. And so that when they're doing those basic functions, doing what they're supposed to, they are just proto-oncogenes. It's when maybe they're switched on, like we see in these examples here, through a translocation, this proto-oncogene gets a different promoter in front of it. So maybe it keeps getting turned on. Now it becomes an oncogene, a cancer-forming gene. Maybe there's duplication of a gene. So instead of having one dose worth of growth factor produced, now there's triple the amount. Here we see a mutation that occurs in a control element here. So again, switching this thing on. Or maybe the product itself gets a mutation in it and becomes much more degradation resistant, or just it is much more active, as a higher activity. So this is why these proto-oncogenes can become oncogenes. Of course, we have to keep in mind that those proto-oncogenes or oncogenes, that's not the only thing driving tumor formation. There is another set of genes called tumor suppressor genes. And so these are sort of counteracting any signals for uncontrolled cell growth. They may be important in regulating how DNA is repaired or sending a cell into apoptosis if there is enough DNA damage that can't be fixed. And so these are all really important. And so when we see tumor formation, it usually means there's a oncogene formation and the loss or mutations within tumor suppressor genes. And here are a couple of examples of where we see mutant cell cycle issues. This one is all about the RAS gene. And so the RAS protein is actually a G protein. If we think back to cell signaling, that's one of these intracellular proteins that's important for transferring the signal. So here we see a growth factor being received. Here's the receptor. It passes that message along to the RAS protein, which through this signal cascade activates this transcription factor and says, okay, yeah, time to divide. And that would be normal cell division in response to a growth factor. Where there's a problem here, a mutation in the RAS protein, see here, there's no signal being received. So no signal has been received, yet the RAS protein is on. In fact, not just on, but hyperactive. So it keeps passing this message along, go, 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 go. And so this transcription factor gets produced a lot, and then the cell divides a lot. And so that's a stimulating pathway. Here's an example of an inhibiting pathway. So here in the normal situation, here we see DNA damage occurring. There's recognition of that by some proteins that would identify DNA damage, and they activate this p53 protein. That will stimulate a response that inhibits the cell cycle and maybe even sends that cell on a path to apoptosis, which is to that coordinated cell death. Here's what happens if, say, p53 isn't working. DNA damage is detected, but that signal can't be passed. It doesn't work. And so p53 isn't activating transcription that would stop that cell from dividing or send it along the path to apoptosis. But instead, those cells continue to divide. And so any damage that's there, it's going to be amplified. And maybe more damage is going to ensue because these checking mechanisms aren't really working, right? This cannot fix or stop a damaged cell from reproducing. All right, so if we think about that p53 protein, which of these options best describes it? All right, so yeah, it's an activator for other genes. It sort of turns on these other pathways that are going to inhibit the cell cycle or send that cell into apoptosis. One of the other options here was that it slows down the cell cycle. It doesn't really do that directly, but it switches on other genes that will inhibit the cell cycle or stop it. So let's put these things together now into what's called the multi-step model of cancer development. And as I've said already many times, you need more than one of these problems to happen, mutations to happen to lead to cancer, and that's why this is multi-step. And so this has been studied in many kinds of cancers, but this example is human colorectal cancers. This is one of the best studied types of cancer. And so here we start to see early on this polyp development, so this little tumor basically. And so this is a result of a loss of this particular gene or something similar to it, this tumor suppressor gene. And so this thing starts to divide unchecked with potential errors, and not really a huge problem at this point. But now what do we see? We see the activation of this RAS oncogene, which we talked about. So now the cells are saying, okay, go, 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 divide, divide, divide. Also, another step, loss of this other tumor suppressor gene. So now multiple things are going wrong. Still benign at this point, so a little tumor here. And then it's not until we start to lose the tumor suppressor activity of p53, and then potentially many other mutations are occurring, that we start to see this thing growing even more and then possibly infiltrating other tissue. This is actually true cancer at that point. And so it used to be that this was called the two-step model. You need one of those oncogenes on and a tumor suppressor off. But it's more widely recognized now. Yeah, that may be important, but you really need probably more than just those things happening. But usually it would involve an oncogene and a tumor suppressor gene mutation. And in many cancers, that RAS and P53 are involved. So that's why we use those as examples, that they often come up in many cases. All right, so that's the end of chapter 16. We'll see you next time.